All right. Hi, everybody, and welcome to Chapter 6, Vision and Chemoreception. So we're starting to get into the sensory receptors that are basically responsible for taking in all the information in our environment and then allowing us to, transduce it into neuronal activity to everything that we see and experience and think about what we see and experience. So, all right. So first, in terms of getting back to sort of a couple of basics and just in, setting our sort of skills or vision abilities and sort of context as well. So the electromagnetic spectrum is actually really, of course, quite large, and it includes all of these waves up here that we are familiar with in various functions, but only in this really small portion right here is this the visible spectrum for us. And, the light waves and all that kind of stuff, that a receptor or anything would be receiving, those are constant, and then we are the ones that actually interpret this spectrum and see various colors. So this is also why animals, will oftentimes have sometimes a smaller, but usually actually a much larger perceptual field in terms of vision, which is based on how likely it is for the success of their survival, the natural selection stuff. And they so they can see much broader, and we'll see colors and things that we cannot physically see, so it's really even hard for us to conceptualize what they look like. We usually have to sort of interpret them somehow. All right, so beginning with the eye, here's a nice graphical picture. So the cornea right here, this is sort of the outer translucent covering of the eye. This bends light when it enters, so about 70 to 80%, so that it can target it to land at, closer to the, land really well within the retina. So it'll kind of basically bend, things so you can still try to get as broad of a perspective as you can down to this smaller space. And the cornea is basically the first thing that light enters when it hits the eye. Then you've got the pupil, which is right here, and this is controlled by colored iris. And this lens right here, this is actually what bends to accommodate light onto the retina. So when you're trying to focus and things like that, that's basically your lens trying to accommodate based upon how far or close you are to things to center the most detailed, important information that you're looking at on the center of your retina. And then your retina is basically all along the back here. And so this whole space, you can see here, it's the yellow. And so you have lots of blood vessels that sort of spin off from this. And this forms your optic nerve. Again, these are these blood vessels. You have virtuous humor in here, and there's a number of really important things that you can tell by looking at eye exams, like risk for diabetes, and or early signs of diabetes, which is even potentially more significant, so someone can catch it. That's type 2 diabetes. And then what the other interesting space is that you can see here where this, where the blood vessels come in, and then you have this little small spot right here where there are no receptors, because there's blood vessels, and that is we actually all have a blind spot. You can kind of do certain things. You can look it up online. I'm going to be, I'm not sure. I don't think the textbook has any looks for this, but you can there's a couple things you can do to like pull the pencil the right way, and you can like see the pencil tip sort of like disappear. But it's so present. Your visual system is actually designed to detect like shifts and change, as we'll get to a little bit later. And so it's so omnipresent all the time that, and it's a small spot that your brain just basically fills in that spot of the visual field with, information from when you move your head and sort of things like that, that it can kind of glean from what would be there. Alrighty, so we're going to go a little bit more into the retina because this is very important. So this is the direction of light actually entering the retina. And so you start at the top, and again, this retina, this little piece of the or a column, shall we say, of the retina. This is just one small piece of it, and it's this whole sort of multi-layers that are all across sort of the retina, and all of these pieces, have to be there for vision to occur. And so one of the important things is once the light enters the retina, and that you can see it's got the ganglion cells and the imicrine cells and the cone bipolar and the rod bipolar cells. And all of these cells have very important structures. But here at the very back are the rods and cones, which are the actual receptors. And so there's technically a fair amount of light that is kind of lost passing through, all of these cellular bodies before actually getting to the rods and cones. I mean, not a lot of light, but a sufficient amount. And but again, it's the receptive fields and of how the rods and cones works and everything like that makes sure that you have sort of like a whole continuous picture. But very much like after wearing, once you're wearing glasses for, quite a while and you get used to them, you really don't sort of see them on your face. Your visual system is really sensitive to change. And when things don't change, they kind of tend to not, disappear, but just sort of like you have to direct your attention to them more, because it's like, oh, well, they're there all the time, so it must not be something significant, because really the visual system generally thinks like, oh, you're moving around all the time, you have to look for a change to navigate this space. If everything's the same, then that's not something that kind of would need to be navigated per se. So so in terms of our vision here, we have phototopic vision, and these are details, bright light, color vision, and these are the cone receptors, these photoreceptors. And this is where you get where you see color. And then the fovea is at the center of the retina, and it's packed with cones. So this would be right here. So you can see this whole little area. This is sort of the fovea, and this right here, that little area. So that's where it's most densely packed with all of the color receptors for cone. So the cone receptors for color, so that you can really see up close and see detail. And that's where you're going to see different shades of color and nuance. And then you can see here, these are the rods. So the rods, they give us scototopic vision, which is vision that's basically geared to detect movement, work in low light, and identify shapes. And so it's, then our visual system is like very efficient, because we have this center sort of detail section, and then we have this outer section that's basically geared to detect things, to direct our attention towards. And so it detects movement and shapes in low light environments, so that we can then try to direct our attention towards them. But sometimes if it's really dark, it's actually directing your attention towards them, will like be harder because then there's not as much color. And so sometimes it's actually easier to perceive an object in the dark if you don't look directly at it and you kind of, try to use some of your sort of like a sort of side glancing vision kind of thing to kind of keep it, but sort of pay attention, sort of also look what's going on the other side. And that actually helps perceive more detail, a little bit more detail in a dark environment where cones are not best designed to function. So all of these cells, so we have our rods and cones, our receptors, and all of these cells basically work with everything together to combine the information that it gets from these various individual receptors to develop and send the information above. And then you're going to have the ganglion cell layer. This is what forms the optic nerve. But there is some like basic kind of processing that goes on in this space where you have really small individual, receptor fields from individual neurons, but then there's so many of them. And how do you start kind of combining that to, to quickly assess our visual field, to quickly assess our visual environment? And one of the ways is actually to have those combinations start to occur much earlier, and so that so you have really early processing. And let's see, I would ask for questions right now. All right, so this is not on the exam, but I just wanted to kind of bring it up here to just sort of point out how it's in the chapter, and basically point out a couple of things that that might be interesting, depending upon your visual experience, whether you wear glasses, whether you're nearsighted or farsighted. And most of some of these visual problems that we have here on the end, on the left, cataracts and macular degeneration, these are things that occur much when you're much older, but this is what some of your parents or grandparents might be dealing with. I know my mom is. So so these are things to just be on the lookout for. The textbook goes into a little bit more detail. One of the things that I thought was pretty neat was I really like how they show, okay, when you have, myopia, you're nearsighted, you can see here that like your eye is a little bit too long. So then what happens is that the space where all the light, everything crosses, that point of like, here's where everything comes together, that should be falling right on the retina right here, is not, and it's off in its space. And then you can see here, if you are, if your eye is too short and if you're farsighted, then then it is falling, like everything is coming together too, like not in time for the retina, too late. And so you're not, then you're not getting, the visual acuity that you would need here or here. And so then the lenses for these conditions are shaped differently. And I thought that like, oh, you guys might, and this is going to be obviously probably not something you'd be able to detect in your glasses, but in the particular lens portion, like how it will try, the ways it will try to bend the light. And so you can see here with this concave lens, then the light comes in and it tries to bend it a little back out to then have it to the point where the information is coming together again right at the fovea on the retina. And then here's the same thing for the convex lens, that it's basically trying to kind of bring together the information a little bit earlier. So again, it can land right on the retina in the fovea. All right, so once the information lands on our rods and our cones, what happens? So the whole process of the, you know, the visual system again is what we have to take from the external environment and represent that information neurally in our body so that our body can process it. And so the process of this is called transduction. And this is the conversion of physical stimulation into nerve impulses. And so here, this this physical, you know, stimulation, you know, like it can be, you know, achieved in many ways. It all depends upon the receptor. But here, this is a basic an example of a rod, and looking at, you know, it at a more cellular level. You can see here it's got its nucleus. Here's its synaptic body, where it's going to try to connect up, you know, with other things. And in here, it's got this inner segment, which is basically the cellular functions. And then it's got this outer segment, that this sort of membrane shelves that are lined with rhodopsin, which is a color pigment. And so, and when the light hits that, then you're going to have like a physical sort of shift or change in the cell in some way. And that will cause the action potential that will then travel down, you know, into the synapse. So here is, so once we know that you have this visual transduction, and then the next thing is like, okay, well, how do we use this to actually see color? So all that my eyes are detecting is wavelengths of light. So what is, you know, so how do we then take this to perceive? Well, of course, you know, you've got your rods and your cones, and they're sensitive, and your cones are sensitive to particular wavelengths of light. So you'll have three different types. And so this is the trichromatic, you know, theory of color vision, and where you're basically trying to perceive the different colors by, you know, combining or processing or looking at the difference between the level of color, or wavelength perception, excuse me, across these different cone types, are short, medium, and long. But one of the things that the trichromatic theory cannot account for is the fact that our visual system can do things, you know, that, like, well, wait, that wavelength thing doesn't really, like, how can it account for that? And so one of them is here, this little sort of test. It's in the textbook, but this is something that you can use, you know, online as well. And I tested it yesterday with my slides on my own computer, but that just means you might have to look a little bit closely. But you want to lean in and put this sort of green spot like right in your center vision, and you want to stare at it, like really, really stare at it, and kind of emblazon it, you know, and then you might actually start to see, you know, around it, like you're looking at this so long, and you can see sort of, and now as soon as you're looking at it for quite a long time and you think, I'm so super sick of this, then you shift your right over to the right, and then you will see this sort of afterglow, shall we say, of a reddish, excuse me, sort of pinkish hue. I saw it and I did it again. And that afterglow is something that the trichromatic theory cannot account for. But you get that afterglow a lot with things, in terms of when you're looking at something for a particular period of time and sort of how it responds. And so since sort of recognizing that, then the researchers developed the opponent process theory. And the opponent process theory is the second theory of color vision, more recent, that assumes cells in the retina and the thalamus send two different signals if they are excited or inhibited. So that's the idea that rods can be excited and rods can be inhibited. Actually, excuse me, I said rods, but this is cones. And so you have you're seeing here, for example, that you've got on the left, the R stands for red, green, yellow, blue. And one of the other reasons that as I'm going through this, that the trichromatic theory cannot account for is why you can't have like yellow blue colors, or red green colors. Like they mix together to make something, completely different. But that's really quite unlike, all the other colors, in terms of these are primary, but like, and the primary has these opposing colors, these contrast colors. But the fact that you can't combine them suggests that this isn't just a wavelength thing. And so this opponent process theory tries to also account for that fact. So this is the idea that for your cones and how they perceive sort of the various colors, and is that you will have a signal like one of the I'm just blanking on the word, guys, I'm so sorry. That one of the it's something better than cones, because it's one of the cones, but it's really sort of the aspects of the transduction process, that the receptor, the word I was searching for sort of more broadly, that the receptor, because it can primarily perceive wavelengths, but then like all of these receptors sort of combine and like work together, as you saw within sort of the diagram of sort of the layers of the retina, that it's not like only the red long cones connect with red long cones, that when you have the bipolar and the immigrant cells, those receptive fields start within the color really young as well. So then you would basically have, through processing this receptive signal from the receptors, that coming down again, forming aspects of the optic nerve to go into the thalamus, is that, well, a neuron can send a signal that basically is this is an excitatory signal for red, and but then green gets inhibited, and which again would combine, sort of like why you can't have those two together. And then when you have an excitatory signal for green, and then Y is inhibited, and then you have the same thing for the yellow and blue. And so this actually ensures that all of the wavelength information comes in for all of these primary colors and our color vision in general, but that there's sort of early, again, opponent processing, where it's a signal just isn't saying like this is red, it's also saying this is red and not green, which gives more information for the visual system to begin processing earlier and help color vision potentially happen much quicker as well, because then the brain's getting two pieces of information, and almost technically a little bit more than two pieces of information when considering the surrounding cells. All right. So this is actually the end of the first section of the chapter. And at the next one, we'll be heading into talking more about these receptive fields and the part of the transduction process. So stay tuned.